Hi students, today we are going to learn about the polyphenolic compounds only, that is of the flavonoids group, particularly on quercetin, kempherol and resveratrol. These are three important molecules which has been studied a lot if you could see the literature. People have seen these molecules 50 years back as anti-nutritional. Now because of the newer research and many of the important issues pertaining to cancer, cardiovascular disease and other chronic degenerative diseases can be prevented using these plant secondary metabolites that are polyphenolic compounds and more so of kempherol, quercetin and resveratrol. These compounds are abundantly present in the plant foods, particularly they are synthesized against the stress and the environmental factors. These compounds are synthesized in the plant foods. These secondary metabolites can effectively scavenge the free radicals and also they can arrest the cancer in the human beings. Many studies have been conducted using polyphenolic compounds and we are learning today about these three compounds, but when we could see the literature about the quercetin, kempherol and resveratrol, these can be like glycoside molecules also, where the glucose moiety is also present along with these compounds. People have shown in vitro studies some of the negative aspects of these two molecules also. These two compounds can act as pro-oxidant and also some of the studies have shown mutagenic effects too. Therefore, there is a need to look into these molecules very clearly in the future studies also, particularly about the resveratrol. This compound has been shown in the red wine. You might have heard about French paradox. French people have less heart attacks comparatively to the rest of the world, and people attributed particularly this polyphenolic compound which is abundantly present in the red wine is preventing these cardiac problems. But these molecules now are also showing some sort of negative effects in some of the studies in vitro. Therefore, now people are going in-depth studies with these molecules. Therefore, we are going to learn these rich sources, their metabolism and ultimately the health effects of these molecules. You will be able to know the rich sources, metabolism, bioavailability and health effects of quercetin. Also, you learn about the rich sources of the kempherol, its metabolism and the bioavailability and health effect. The third molecule, the resveratrol, the important food sources, the resveratrol metabolism and the bioavailability of resveratrol and the health benefits of these three molecules you will be able to understand from this module. So, let us start with the important molecule, the quercetin. What is quercetin? Quercetin is a type of a polyphenol belonging to the flavonoid class. As I have mentioned, this is the polyphenolic group, secondary plant metabolites synthesized in the plants. Over 4,000 flavonoids have been discovered so far and twice as many polyphenols have been identified. They are widespread constituents of many plant foods including fruits, vegetables, grains, chocolate, tea and wine. Flavonoids are the most abundant antioxidants in the human diet and they modulate the activity of a wide range of enzymes and cell receptors. If you recollect that human cells have constitutively present cellular antioxidant enzymes. When there is more free radical load, you need to supply antioxidants from exogenous sources. Plant foods are rich sources of these antioxidants flavonoids, and particularly the quercetin molecule, which is abundantly present in berries, apple, onions are the rich sources, and particularly even the other fruits and vegetables. For example, if we consider apples as a source of antioxidant polyphenols, apples are widely consumed across the globe. India is also having many varieties of apples produced, rich source of dietary antioxidants. In addition to that, the important molecule which we are discussing is the quercetin is also present in the apples. Suppose you take 100 grams of apple, have the same antioxidant activity as 1500 milligrams of vitamin C. That is, what I am trying to tell you is that if you take 100 grams of apple, which is equivalent to 1.5 grams of vitamin C. The average apple weighs approximately 150 grams. Apple peels contain from 2 to 6 times more phenolic compounds and 2 to 3 times more flavonoids than apple flesh. Apple peels and whole apples are better able to inhibit cancer cell proliferation in vitro when compared to peeled apples. What we have to learn here is that quercetin, an important antioxidant, a flavonoid which is abundantly present in the apple, having higher concentrations in the peel portion. But some people, they remove the peel and they will eat the flesh only. But the advantage of eating along with the peel have an added advantage for cancer cell proliferation inhibition and prevention of cancer. So, how the quercetin molecule is present in the natural form? Quercetin is present in foods as quercetin glycosides. Apples contain several quercetin glycosides: quercetin galactoside, glucoside, arabinoside, rhamnoside and xyloside. These are the glycosides where the quercetin molecule having this moiety in the structure. The type of sugar moiety influences bioavailability and metabolism. This is what we have to understand. See, when you talk about a quercetin, it is an aglycon without glycoside. But when you have the glycosides which I have mentioned just now, either about the arabinoside or rhamnoside or xyloside, the metabolism, the bioavailability influences because of this structural difference. Quercetin glycosides from apples are absorbed similarly to purified quercetin 3 rutinoside, which is also called as rutin. We look into the metabolism. Quercetin glycosides are cleaved before absorption and then rapidly metabolized into quercetin glucuronides and sulfates. See, when in the natural form, as I have told you, these glucosides are present. When once it is in the digestion process, before absorption, these glycosides will be cleaved and the aglycons will be absorbed efficiently. Quercetin is conjugated into glucuronides and sulfates by intestinal cells and the liver. See, once it is absorbed, again it will be conjugated or sulfates in the intestinal cells and also in the liver. Most of the circulating plasma quercetin existed in conjugated forms only. Very little quercetin in aglycon form is found in the circulation. That means the natural form, the quercetin glycoside, once it is cleaved and absorbed, it will get conjugated, and most of the quercetin conjugated forms are only in the circulatory levels. Quercetin glycosides as found in apples, for example, are not found in the circulation, means they are actually cleaved. Once they are absorbed, they will get conjugated in a different form. Now, we will look into various foods and beverages which contains these quercetin in appreciable quantities. I am giving here that more than 1 milligram per serving of these fruits, vegetables, grains and beverages category. For examples, in the fruits category, the rich sources of quercetin includes apples, apricots, blackberries, blueberries, cherries cranberries, currants, elderberries, grapes, plums, raspberries. In the vegetable category, green beans, broccoli, celery, collard greens, kale, onions, spinach, tomatoes. Grains, particularly buckwheat and kasha. In the beverages, black tea, green tea and the red wine are the important sources for this quercetin molecule. What are the epidemiological or cohort case control studies indicate about the quercetin? That habitual consumption of apples, onions and other dietary quercetin sources may reduce lung cancer risk approximately 50%. So, it has been indicated in this epidemiological studies that quercetin is a molecule that can reduce the lung cancer very effectively. And if you could look into the literature of many of these in vitro chemopreventive actions of quercetin, many of the studies which are published have shown it prevents the free radical mediated DNA damage. It regulates carcinogen metabolism. Also, it inhibits proliferation and induces apoptosis via numerous mechanisms. So, what is the clinical data is telling particularly on the human trials? Purified quercetin and rutin increases resistance of LDL oxidation, improve prostatitis and cystitis symptoms. No adverse effects noted from daily doses up to 1000 milligrams for 30 days in the clinical trials. Quercetin rich foods increase resistance of lymphocyte DNA to strand breakage and ex vivo oxidative insult. Increases plasma antioxidant capacity, the quercetin rich foods. Also increases red blood corpuscle superoxide dismutase activity also. So, if we look at the pharmacokinetics of the another important molecule which we are going to discuss now about the kempherol. So far we have discussed about the quercetin. Now we will learn about the kempherol, its pharmacokinetics. When the kempherol rich foods once ingested orally, which it has kempherol or sometimes as kempherol glycosides. Once they are cleaved and entered into the small intestine where the metabolism takes place, and it will be absorbed in the circulation and it will enters into the liver both as aglycan as well as the glycoside. And then once this is cleaved and it can be stored and it can be given to the other tissues also as kempherol conjugated forms. This can be collected from the tissues also, and once it enters in the systemic circulation, they will be excreted through the kidney into the urine. This is the way the pharmacokinetics of kempherol takes place. In a study recently published about the kempherol effects on cell viability, if you look at from 0 to 100 micromolar, the cell viability has been taken care, particularly when they have studied about the matrix metalloproteinase 2 expression by downregulating ERK1/2 and the activator protein 1 signaling pathways in oral cancer cells. Further studies have also shown the kempherol suppression of MMP2 expression at a transcriptional level and on DNA binding activity of AP1 in SEC4 cells. It has been shown that it is very effective, particularly in DNA binding and at the transcriptional levels also. So, therefore, the kempherol has been shown at cellular level that it can effectively work against the cancer. Now, the third molecule which we are going to learn about, resveratrol. This resveratrol compound belongs to a class of polyphenolic compounds called stilbenes. Some types of plants produce resveratrol and other stilbenes in response to stress, injury, fungal infection and ultraviolet radiation. It is a fat soluble compound that occurs in a trans and a cis configuration. So, you should understand that resveratrol molecule can be in both trans as well as cis configuration. Both cis and trans resveratrol also occur as glucosides bound to glucose molecule. Very important in a natural form, where usually many of the compounds are trans compounds which are tagged with these glucose molecule, that is with the glycosides, but whereas even the cis forms of resveratrol have also been shown to have glycosidic bonds in their moieties. Resveratrol 3O beta glucoside is also called pisceate. Resveratrol is a phytoalexin, a class of antibiotic compounds produced as a part of a plant's defense system against disease. Very important information about resveratrol is it is also called as phytoalexin. Earlier I have mentioned about the group of phenolic compounds called stilbenes also. Now you have also learned that resveratrol is a phytoalexin, a class of antibiotic compounds. For example, in response to an invading fungus, resveratrol is synthesized from paracoumaril CoA and malonyl CoA. When there is a fungus attack on a plant or on a tree, this compound is synthesized to counter the fungus activity. Since fungal infections are more common in cooler climates, grapes grown in cooler climates have a higher concentration of resveratrol. Very important point which we have to understand. The stress induced or fungal induced stress to the plant can be easily encountered through the synthesized molecule like resveratrol in the grape, which has been shown particularly in the cooler climates, these compounds synthesized in higher concentrations. What are the food sources of this resveratrol compound? The amount of fermentation time a wine spends in contact with grape skins is an important determinant of its resveratrol content. As I have told you earlier about the apples and the quercetin, which are abundantly present in the peel than the flesh. Here also, if you take particularly the grape skins are rich sources of resveratrol. While wine preparation, that is a fermentation when it is taking place, when the skins are in contact, therefore there is an every possibility of more resveratrol in the wine. White and rose wines generally contains less resveratrol than red wines, which is because the skins are removed earlier during their production, lessening the amount that the resveratrol which is extracted. So therefore, what we have to understand, the external part of the plant or the fruit having more of these polyphenolic compounds for the defense mechanism, which we should utilize this as an antioxidant molecule, and therefore we should not peel the fruits, where we have to understand from this process of fermentation where the red wine contains more of resveratrol comparatively to the white and rose wine. Red or purple grape juices may also be good sources of resveratrol. So we will look into the content of the resveratrol in the wines and the grape juice. White wines, this is Spanish variety, resveratrol milligrams per liter 0.05 to 1.8, and rose wines is 0.43, whereas red wines, if you could see it, very high 1.92 to 12.59 milligrams. Even the red wines global also are rich sources of resveratrol comparatively to other rose wines. Red grape juice Spanish also contains very rich source of resveratrol up to 8.69 milligrams per liter. So therefore, choosing such molecule from the nature and the food source is going to help particularly to combat the chronic degenerative diseases. The other foods also, in addition to those grapes and apples and juices which we have discussed, particularly like peanuts either raw or boiled, peanut butter, and red grapes also contains quite good amounts of resveratrol. For example, peanuts are rich sources of resveratrol comparatively to the peanut butter. So, metabolism and bioavailability of resveratrol. Although trans resveratrol appear to be well absorbed by humans when taken orally, its bioavailability is relatively low due to its rapid metabolism and elimination. Information about the bioavailability of resveratrol in humans is very important to understand the human health benefits. This is because much of the basic research on resveratrol has been conducted in cultured cells exposed to unmetabolized resveratrol at concentrations that are often 10 to 100 times greater than peak concentrations observed in human plasma after oral consumption. This is what we have to understand. We should first know the sources, their content, the quantity which could be bioaccessible and bioavailable. Such quantities only if we do in the cell culture or in human studies, that is more relevant. That is what we have to understand. So, therefore, these compounds are very well established against the chronic degenerative diseases. Significant reductions in cardiovascular disease risk have been associated with moderate consumption of alcoholic beverages, particularly which are the sources of these resveratrol like red wine. As I have mentioned initially, people talked about French paradox because of this molecule. Red wine contains resveratrol and even higher levels of other flavonoids also in addition to resveratrol. These polyphenolic compounds have antioxidant, anti-inflammatory and other potentially anti-atherogenic effects in the test tube and in some animal models of atherosclerosis. Although moderate alcohol consumption has been consistently associated with 20 to 30% reductions in coronary heart disease risk, it is not yet clear whether the red wine polyphenols confer any additional risk reduction. In the cardiovascular disease area, resveratrol has been found to exert a number of potentially cardioprotective effects in vitro, including inhibition of platelet aggregation, promotion of vasodilation by enhancing the production of nitric oxide, inhibition of inflammatory enzymes. But concentrations of resveratrol required to produce these effects are often higher than those that have been measured in human plasma after oral consumption of resveratrol. Although its presence in red wine has stimulated much interest in the area of cardiovascular disease prevention, currently there is no convincing evidence that resveratrol has cardioprotective effects in humans, especially in the amounts present in 1 to 2 glasses of red wine. In the cancer area, resveratrol has been found to inhibit the proliferation of variety of human cancer cell lines, including those from breast, prostate, stomach, colon, pancreatic and thyroid cancers when added to cell cultures outside the body. In animal models, there has been marked inhibition in the development of esophageal, intestinal and breast cancer with oral administration of resveratrol. In mice genetically predisposed to colon cancer, effects from oral resveratrol administration have been mixed results. It is currently not known whether or not high intakes of resveratrol can help prevent cancers in humans. Studies suggest that even very high dietary intakes of resveratrol may not result in tissue levels that are high enough to demonstrate the protective effects that resveratrol has shown in cell culture studies. What does it mean? It means the bioavailability is really crux of the problem. So, the longevity. Caloric restriction has been proven to extend the lifespan of number of species including mammals. In yeast, a caloric restriction stimulates the activity of an enzyme referred to as SIR2. Administering resveratrol to yeast increasing SIR2 activity in the absence of caloric restriction and the extending the replicative lifespan of yeast by 70%. Although resveratrol did not increase the activity of the homologous enzyme SIR1 in the test tube, whether or not resveratrol can extend the human lifespan is currently unknown. And again, the resveratrol concentrations that were necessary to increase SIR T1 activity in the test tube were considerably higher than any concentration previously measured in human plasma. Reasons why recommending a population-wide increase would be premature. Little is known about the absorption and clearance of resveratrol. This is what we have to understand. Any molecule for that matter, and here is the resveratrol. The identities of its metabolic products or its effects on the liver. The research on resveratrol has focused on its short-term effects and has been dominated by in vitro studies and non-human models. That is the weakness. Its role as a potentiator of breast carcinomas may significantly limit its use even for its proven benefits. Its main dietary source is red wine, which is not only extremely variable but possibly harmful, to be recommending increased intakes of red wine to the population at this point. At present, relatively little is known about the effects of resveratrol in humans. In conclusion, flavonoids and flavones, which widely distributed in the plant kingdom, epidemiological studies have found a positive association between the consumption of kempherol, quercetin and resveratrol containing foods and a reduced risk of developing cardiovascular diseases and some types of cancers. Numerous in vitro and some animal studies support a role of kempherol, quercetin and resveratrol in the prevention and or in the treatment of these and other diseases such as neurodegenerative diseases, infectious diseases, diabetes, osteoporosis, anxiety, allergies, inflammation and also in pain. However, many of these studies have been conducted at doses higher than those documented in humans, and therefore, it is very difficult to predict from these results the effects of kempherol, quercetin, resveratrol and intake on the prevention of these diseases. Additional animal and human studies and clinical trials are needed to better understand the possible health effects of kempherol, quercetin and resveratrol and to further evaluate its potential use as a new drug or the new molecules which can inhibit or prevent the cancer and other chronic degenerative diseases. Of all the things, what we have to learn from these polyphenolic compounds are their bioavailability is very important. Usually these polyphenolic compounds are very poorly absorbed. Therefore, their effects, how it is going to be for those levels of absorption and those levels of circulation, one has to really look into. However, consuming these important foods like what we have mentioned about apples, grapes and many other grains or which are rich sources of these flavonoids, particularly of these kempherol, quercetin and resveratrol, will certainly help as antioxidant molecules in the prevention of many of the cardiovascular, cancer and the diabetes and other diseases too. Thank you.